good. Well fed and healthy. I see a lot of smiles. Yeah, no, but anyway, so we're lucky to have Dr. Jim McKerrow, who many of you know already from I3. And every time he gives his talks during I3, I always like, you know, I have to keep myself down from asking him about this work that he's doing, which is really the bulk of his creative discovery work at UCSF. And I think, you know, he has a lot of accomplishments, of which I wouldn't summarize here, but the main thing that's really impressed me about Jim is that he puts science into social context more than a lot of people do here, and a lot of people do that here, and that's always a great model for all of us to do. So without further ado, thanks a lot.
Okay, thanks. So I'm going to talk about something which I think is very relevant to translational research, and that is how do you develop drugs for what are called neglected tropical diseases. So I think all of you probably have some sense of what a neglected tropical disease is, but just as a reminder, we're talking about diseases that affect hundreds of millions of people worldwide. I'm going to be focusing on those diseases that are caused by parasitic organisms. So, for example, schistosomiasis affects 250 million people. African trypanosomiasis is a huge problem in sub-Saharan Africa, particularly in East Africa. If any of you have ever been there, this is what's spread by the tsetse fly. But the problem is, even though so many people are affected with these diseases worldwide, we currently really don't have effective drugs, or if the drugs are effective, they're not safe. And if they're effective and safe, then we have a problem with drug resistance. And for all of these diseases, I'm just showing you 4 here, there's many more. The problem has been that we really don't have any new drugs really in the last 50 years or so. And that seems kind of odd. Why would you not have drugs to treat diseases that affect so many people? And I think most of you know that the issue here, the major issue, is that there's little interest in the pharmaceutical industry in drug development. And it's the pharmaceutical industry worldwide which is really responsible for making new drugs. And the reason I think most of you are also aware of why there is little interest in the pharmaceutical industry is even though these diseases affect hundreds of millions of people worldwide, they tend to be poor people in resource-poor areas of the world. So there's really no market incentive for pharmaceutical companies to get involved in this. And I'm going to be telling you a little bit about different approaches to take to try to overcome this issue, which is a major issue. But I want to emphasize that you can interrupt me at any time. We can kind of go off on a tangent and talk about things like, well, you know, how many drugs did we get last year from the pharmaceutical industry, or anything else you have to be interested in. So the big issue in not having the pharmaceutical industry involved is that drug development, by and large, is driven by industry. So if we talk about what's done here, for example, at UCSF or at UCLA or at UCSD or any of the universities on the East Coast or basically any nonprofit in the world, it's basically this first box right here. And what companies would call this is target family selection. What we would call it is basic or discovery research. Okay? So this is the kind of research that's done, you know, within 100 feet of where you're sitting right now, which would be studying the basic biology of these organisms, studying their biochemistry, perhaps even identifying weak links in the way that they infect the human host. But all of that is basic. And everything else in this so-called drug discovery pipeline is something that would normally be done in industry. So after you recognize a target in a specific organism, and this might be usually is a specific molecule, so it might be an enzyme, it might be a receptor, it might be a surface protein. The next step is usually in a company to develop a large number of chemicals that would hit that target, inactivate it, and therefore block the ability of whatever that infectious organism is to replicate. So all of this chemistry which is involved here would be done in industry. And there's a lot here, not only in terms of the initial identification of so-called hits against this target, but also the development of those hits into something that can be given to an animal model of infection, for example. So that means that it's not only a chemical that hits your molecular target, but it has to be safe in mice, or whatever you're using as your experimental model. It has to get in and get into the bloodstream and get to the tissue where the organism is in reasonable concentrations. So that involves a whole separate chemical development that's going on in parallel to the chemical development that's going on to the target. So there's a lot going on here. Then, even after you're done with that, you have to show that your lead compound is now safe and also efficacious if given like a drug. So what's the usual way we take a drug? We just take a drug by mouth. So in industry, that's called oral bioavailability. So you have to show that your chemical that's hitting this target is orally bioavailable and is safe. And our Food and Drug Administration in the United States, and this is also true of the one in Europe, requires that you carry out what are called preclinical studies in at least 2 other animals beyond rodents. So that usually means the dog, legal dog, and quote, NHP, non-human primate, which is like a little monkey. Okay, so the FDA requires that you give your compound to dogs and or monkeys, show that it's safe at the levels that you want to give it to people, and show that it gets in and it has a reasonable half-life, that it lasts in the body for a long period of time. And you have to do all of that before you're ever allowed to then test this in people. So the next step is called phase 1. Phase 1 is basically a safety study. If you were doing a phase 1 study with a cancer agent, you might actually do that on patients with cancer and see whether or not it's safe. But in terms of these diseases, you don't have to obviously use people who are infected. This can be done here in the US, even though the diseases are primarily elsewhere in the world. And what you would have is a group of what the FDA would call normal, healthy volunteers, right? Think about that for a minute. Do you know anybody who's normal and healthy? No, probably not, right? So you know, when I went to the FDA, I said, well, what do you mean by normal, healthy volunteers? And they said, well, you know, like medical students. So when you go around here and you see on the bulletin board, there's like a little thing that says, earn $600 in a drug study, and there's a little like tear off cell phone number. That's exactly what this is. It's a phase 1 study, and they're trying to recruit you by paying you a fair amount of money. So what happens there is you're given a very low dose of whatever this new drug is, and you're monitored for 24 hours to make sure it's safe, and then your blood is drawn to see whether it got into your bloodstream at the same level that would be predicted from doing this study in a small monkey or a dog. Okay? So if it's safe and if the so-called pharmacokinetics are appropriate, then you're allowed to go on to what's called phase 2. And phase 2 is really the first time that you're testing this drug against people who have the disease. And in that particular case, you would have to do this, if we're talking about these neglected diseases, somewhere else, right? So for example, if you were doing a study of African trypanosomiasis, you'd have to do that in East Africa. If you're doing Chagas disease, you might be able to do it in Brazil. If you're doing amoebiasis, you can do it in Mexico City. And what happens in phase 2 is that all you want to show is that your drug has some effect on the course of that disease. So this is not a double-blinded study. This is basically pick, say, somewhere between 20 and 50 people who are infected. You treat them, and then you have some parameter that you can follow, okay, which tells you that your drug had a positive effect. So what's that parameter? Well, you can imagine if this were a cancer study, it might be a radiologic evaluation of the size of the tumor in someone's lung, and you see it shrink. Okay, that's what you would do in phase 2 for cancer. For an infectious disease, if you can actually measure the number of organisms in the bloodstream, that would be great. Or if you have some surrogate marker, maybe a ELISA test, some kind of serology, or perhaps some biomarker that you could follow to show efficacy, that would be fine as well. And only after you've shown efficacy in patients you know have the disease are you allowed to then proceed to phase 3. Phase 3 is what you usually read about in the paper. This is the huge study with thousands of people. You have to have a statistically significantly large population. It's all double-blinded. You don't know whether or not you've really done something until the very end when you break the code. And then you present that information back to the Food and Drug Administration, and if they consider it effective and safe, then they will approve that drug for what companies would call file and launch. And the cost of this whole process is at least $500 million. Some companies would argue it's a lot more. Okay? And as a result of that, the reason that companies are not interested in the diseases we're talking about is they're never going to make back the $500 million that they invested in this process. So today, as you might imagine, companies are looking for what are called blockbuster drugs. Okay, that's a drug that your company will tell you, if they go out and they market that, they will make $1 billion. That's a blockbuster drug. You have to make $1 billion, right? So think about, okay, what diseases could I treat where I would get $1 billion back? Well, if you think about infectious diseases, probably not, because you're talking about things like antibiotics, which you take for, and all of you have probably taken antibiotics, what, 10 or 20 days at the most, okay? If you put that into an economic package and you calculate, even if you charge $10,000 for a course of that therapy, you're never going to make $1 billion. So forget about infectious disease. What companies are looking for now for the blockbuster drug is not a disease you cure, but a disease you manage. Okay? So you want to be having patients take this drug for the rest of their lives. So what would that be? Okay, yes, cholesterol lowering, high blood pressure, asthma and allergies, okay, things like that, where you would manage the disease, but you always be taking the drug. And these diseases do not fit into that category. And the other thing that you may be aware of is that as a result of the fact that you have only these blockbuster drugs now, which are really the focus of industry, and there's been all this merger. You've read about this in the paper. You know, like Pfizer bought Upjohn. Okay, so Upjohn doesn't exist anymore because it was bought by Pfizer. Everything has been consolidated into a few companies, and all the companies are looking for the blockbuster drugs. So CEOs of these companies will admit to you, no problem whatsoever, that they actually are not investing a lot in research anymore. What they're doing is they're investing in the legal side of their company to do what they call patent busting. And that is, they go out and they say, well, I need a blockbuster drug, and I think all the good blockbuster drugs have been made, but let's see if we can get around this company's patent. So let's take Lipitor, for example, the cholesterol lowering. Let's have our patent attorneys look at the structure of Lipitor and see, oh, gee, if our chemist put another methyl group on here, will that get around that patent? Okay? And they'll just admit to you, look, this is what we're doing. This is where we're putting our investment. And so that's why you see, unfortunately, so few new drugs. We talk about novel chemical entities. The FDA calls these NCEs, novel chemical entities. There's maybe, last year, I think there were four out of all the companies in the world, 4 novel chemical entities came out. Everything else was sort of the same, different formulation, combination, or slightly different. Okay, so given all of those issues, then how can we approach trying to come up with new drugs for these so-called diseases? And I'll show you 3 different approaches that people are taking. They're taking here at UCSF, but I'm just going to give you this as an example, and you probably see the same thing happening in a number of other places around the world, both academic institutions as well as a number of nonprofit institutions. Okay, so the first is what's called target-based or cell-based screening. So what are we talking about here? Well, if we're talking about a cell-based screen, what we're talking about is the kind of thing that used to be done in companies, but now is pretty much farmed out. And that is, if you, again, we take the example of a new anti-cancer agent, you might have a cell line from a colon cancer in your laboratory. And you plate that cell line out into what are called microtiter plates. Those are little plastic plates with all over the wells, and some of them may have dealt with these. And those can have a lot of wells, and you can have 384 or even more wells per plate. So you plate out your cancer cells in that. And then you use some type of robotics to rapidly put in each of those wells a different possible chemical that we think might be a drug and see whether it affects the replication of those cancer cells. So the same thing can be done theoretically with pathogens. Okay, you can see, well, obviously bacteria, they're nice and small, but you can even do it with some of these parasites, and I'll show you how that's put together. So in this case, instead of a cell-based screen, you might call this a pathogen-based screen, but it's the same idea. Okay, so what you're doing is you're creating a situation where you've adapted some kind of assay to a microtiter plate format so that you can then use robotic liquid handling to screen through a very large number of compounds. And I'm going to give you an example of a specific phenotype that's come out of this pathogen-based screen that then suggests a molecular target. Okay? Even though you're starting out by directly screening against the pathogen itself, you wind up identifying a molecular target. And that's great, because now you can go after that molecular target a lot easier than you could after the whole pathogen. And the FDA loves it when you know what the mechanism of action of the drug is. Having a molecular target is very important. Okay, so basically what we're talking about here is screening what I call big libraries of small molecules. So for those of you who aren't chemists, a library refers to a large number of chemical compounds. And small molecules refer to chemicals of less than 600 molecular weight. So why is that important? Well, various investigators have asked the question, if we take all the drugs that we have, okay, and we ask, what are the qualities of those drugs? One of the qualities that comes up a lot is that many of them are of small molecular weight. And that's important because it turns out that small molecular weight compounds don't get turned over by your body as rapidly, so they stick around longer, and they're also absorbed better. Okay, so those are probably the two main reasons why small molecules became very popular. The other reason is that companies, and these are a few of the ones that have collaborated with people here at UCSF, for example. Companies have found that they can utilize a new type of chemical technology called parallel synthesis to start out with one small molecule and then make a whole lot of derivatives of that small molecule very rapidly. Okay? And that can also be done basically robotically. And so it's very easy for companies now to create very large libraries of small molecules. So what are big libraries? I'm talking about, in some cases like Novartis, libraries that are in excess of 1 million compounds. That's a lot, okay? But remember that those compounds are synthetic, and they tend to be derivatives of similar things, all right? So they're huge libraries, but in the end, they're not as diverse as you think they might be, and that turns out to be a problem for companies. So in order to carry out this kind of screening, the real key is that you have to adopt your screen to robotic liquid handling. And so here at UCSF, this is done over on the Mission Bay campus in a center called the Small Molecule Discovery Center. If you're interested, they have a website you can look at. This is sort of a snapshot of one of the rooms. This is a room that happens to be at a level that one can do pathogens in it. And basically what's involved in all of this is, first of all, you have all of your compounds in deep well plates that can be loaded into these kind of cylindrical towers that are called hotels. Okay, so you can load, say, 10,000 different compounds on plates in these hotels. And then back here, there's a robotic arm which will go and grab one of these plates out of the hotel, bring it over and put it on this machine. And this machine is a robotic liquid handler. So what it will do is it will add the appropriate media to that plate, and then it'll add the appropriate parasite to that plate. Okay, so all of what you would want to assay, the chemicals, the media, and the parasites, can all be done robotically. Then the arm takes this and brings it over a little bit off stage here to an incubator. You'd incubate it so that you can see whether that compound is affecting parasite replication, for example. And then there has to be some kind of readout, okay? And because you're looking at robotic liquid handling, the readout has to be something which can also be automated. And that generally means that some kind of assay which you have a fluorescent readout or a luminescence readout or an image analysis readout. And so this is the hardest thing. It's developing the assay for whatever you're looking at that has a readout that can also be automated. If you do that, you can really screen hundreds of thousands of compounds in a week.
So how much would a robotic sample cost?
Okay, so what does all of this cost? Okay, each of these instruments is probably hundreds of thousands of dollars. So this little room here is in excess of $1 million, which is why there's only one of them on campus right now. Okay, a company might have an even bigger room. In fact, it's kind of interesting. Novartis down in San Diego has a screening facility like this, which they use for various things. What they did is, if you remember Saturn cars, okay, well, they don't exist anymore, but what Novartis did is it went to the factories where they were building Saturn cars and it bought very cheaply, wholesale price, all the big yellow robotic arms that they were using to make the cars and then adopted them to their system. So they have like a warehouse with, 5, 6 of these. So if you can afford that, yeah, this is the infrastructure input is very expensive. And then of course you have to have the expertise to run this, and then you also have the expertise to repair it. Because one thing to have the equipment is another thing to keep it maintained. Last month I was down in Brazil, and people in Brazil have been very good at getting grants to buy equipment like this. They set up the equipment, in a couple of months something breaks down, and here at UCSF you just get on the phone to the technical representative from that company who happens to be in the Bay Area, because there's like, 100 other of these instruments in the Bay Area, and they'll be over maybe the next day. Oh, you might have to wait 2 days. In Brazil, there is no one, there is no technical representative in Brazil. Oh, there's no technical representative in South America. Okay, so there's no way they can get that repaired. So either you know how to repair it yourself, or it just sits there for like forever. So you're right, it's a big problem. Okay. So let me show you an example of this in terms of each of these organisms. So each of these organisms has now been adapted to that kind of format, so you can do robotic liquid handling. And that's kind of cool because these range from single-cell organisms like Giardia that some of you may have gotten infected by, all the way up to things like Trypanosoma brucei. This is the organism that causes trypanosomiasis, and then these really bizarre multicellular worm parasites as well. So all of these have been adapted. You can see that the difference in these different screens depends upon the size of the organism. So some of them you can have pretty high throughput, like this guy and this guy, and some of them, you can't do hundreds of thousands of compounds. You can maybe do tens of thousands of compounds. Okay, so then you have to be a little more selective in what you screen against, but it is possible. So the first assay, and the simplest one, is this one for a single-cell parasite that lives outside of cells. Just to show you how this assay was developed, if you want the details, you can read about it here. It turns out that this single-cell protozoan in a microtiter plate generates ATP, and that's very important because it turns out that ATP generation by this parasite is a good measure of its viability. So if you don't have any ATP generation, the parasite is dead. Okay, so there's a very nice correlation. So what Zachary Mackey did was to couple ATP generation to the luciferase reaction, which is what the firefly uses to make its light, okay, to produce luminescence, which can be measured automatically, okay, and then that became the assay. It was a luminescent assay for viability of Trypanosoma brucei, and this is now allowed hundreds of thousands of compounds that are screened against this organism, which causes African trypanosomiasis. A more difficult situation would be an organism that lives inside of a human cell. Okay, so this is an extracellular situation, something where now you have the complication of of a parasite, in this case Trypanosoma cruzi, or this could be leishmania. This causes Chagas disease, this causes leishmaniasis, and they replicate inside of human host cells. So in this particular case, instead of using a luminescent output, one uses an automatic imaging system. And the one that's over at Mission Bay is called the InCell Analyzer 2000. There are several instruments like this. And what it does, it's like an automated microscope. It looks at each of those little microtiter wells, takes a snapshot that goes into, that gets digitized, goes into a computer system, which then you can write code to analyze it in different ways. I'll show you what's done here. And all of that can be done in about 150 milliseconds or even faster. Okay, so you can see that you can really move the number of wells through this instrument very quickly. So what the instrument does is it distinguishes between parasites replicating within the host cell and the host cell movements. And it gives you two readouts. How many parasites are in the host cell, how many host cells are infected, and whether the drug lead that you put in had any effect on the host cells themselves, which you don't want, right? So what you really want is something which kills the parasite and doesn't kill the host cell. So this is actually kind of a cool assay because even though it's a little more involved than the one I just showed you, it gives you two different pieces of information. Okay? So this has also been published a couple of years ago. I won't go into it. Okay, so then the third situation is probably the most complicated, and that's when you start dealing with these multicellular parasites, these parasitic worms. And there's many. There's lymphatic filariasis, river blindness, schistosomiasis, etc. So this is the one for schistosomiasis. And the assay was put together in a collaboration between Raul Singh, who's the chair of computer science at San Francisco State University over there, and Connor Caffrey and Brian Suzuki here at UCSF. And so what Raul's group did was they wrote a cart decision tree, which is basically computer code, to identify the effect of chemicals on these worms, because even though these worms are kind of large, so the adults grow to be about 6 millimeters long, okay? Even though the worms are rather large, and so you're limited in the number that you can look at, nevertheless, there's an advantage in that they'll have different phenotypes depending upon the drug that they're exposed to. So it turns out that calcium channel blockers produce this sort of blow up of the worm. This is the drug that's used now, praziquantel, kind of blows it up in the middle. These are inhibitors of proteases produced by the parasite that become this granular thing. And then this is normal, okay? So you can see what this cart decision tree does is it kind of lumps together these phenotypes. So if you come in with a new chemical compound that kills these parasites, sometimes you can make a pretty good guess at what the molecular target is based upon the phenotype. And this is true not only for shape, but also for movement. So this is what an adult schistosome parasite looks like when it's moving around in the well, okay? So it's pretty big. This is the male. That's the mouth, which is eating your red blood cells. It has a little sucker here that can suck onto your blood vessels so that it can stay there against blood flow. It can crawl around like an inchworm. I just want to give you an idea of the speed of movement. So this is in real time, okay? So here's another well in which some chemical has been put in. And you can see immediately that what's happened now is the worm is moving faster. Everything's moving fast, and then every once in a while you see this kind of weird peristaltic thing go down the length, where there it goes, okay? And so, you know, if you've ever worked on Drosophila, all the Drosophila people have these cute names for their genes. So we thought, hey, come on, can we have a cute name for this effect? So we decided to call it schistodisco. So what's causing schistodisco? And it turns out that all of the compounds that produce this schistodisco phenotype were inhibitors of an enzyme called phosphodiesterase 4. And that turned out to be kind of cool because phosphodiesterase 4 turned out to be the enzyme that turns over cyclic AMP at the neuromuscular junction of worms. And it turns out that not only do parasitic worms have phosphodiesterase 4, but everybody's favorite developmental experimental model, C. elegans, also has a phosphodiesterase 4. And so it was from the C. elegans work that you could figure out why you were getting this hyperactive phenotype, because if cyclic AMP is the signal from nerve to muscle, and this turns over cyclic AMP, and you inhibit it, you get more cyclic AMP, you get more signal, you get more